Good morning or good afternoon. Hey guys, and welcome to another fun lesson about the kingdoms of life. We're going to go ahead and dive in with some more detail to the two prokaryotic kingdoms of life, eubacteria and archaea. And this should help us to understand sort of the evolutionary basis for eukaryotes, as well as an idea of what early Earth looked like and the first kind of life that roamed its surface. So let's go ahead and dive in. About 3.6 billion years ago, the first life appeared on Earth. Now, at that time, the Earth was very different than what we see it as today. Volcanoes were erupting, the ground was heaving, oceans had newly condensed, newly formed because the Earth had finally cooled enough for water to actually not just be vapor, but actually be liquid. And it was starting to spread across the surface of the Earth. And within everything, the water and the rock and all the stuff that was going on in the atmosphere, there were these molecules that were interacting with each other, these chemical molecules, and eventually they formed into the macromolecules that you guys know of, are carbohydrates, lipids, nucleic acids, and proteins. And when they did that, they started to form together in more and more complex ways. Evolution and natural selection actually worked to change these very early forms, even before life began. And eventually, the first cells emerged from this teeming mass of chemical interactions. And those first cells that emerged were what we know as prokaryotes. Bacteria or eubacteria were the first ones out of the gate. When they evolved in a very simple form that was highly effective for survival. They had a cell wall for protection. It was made out of a carbohydrate called peptidoglycan. They had a cell membrane like the one that we would recognize today, looking at our cell membranes, a lipid bilayer just beneath the cell wall. They had a flagellum for moving around. They had cilia and pilus, these little bitty, like the little tentacles and hair sticking out of the side for sensory things and movement. And they did have DNA. They didn't have a nucleus or membrane-bound organelles, so they didn't have any of the complex stuff we would recognize in a eukaryotic cell, but they did have DNA and they had ribosomes. And that was how they made their proteins and did all their translation and transcription. They were able to survive in an incredible number of habitats because of this very simple yet diverse form, and they expanded, adaptively radiating to all the various niches that were available. In shape, bacteria typically diversified into three, though there are more shapes. The three most typical shapes for bacteria were bacillus or rod-shaped, coccus or cossi, spherical shaped, and spirillus or spirillum, which was the spiral. And so these three different shapes of bacteria allowed them to move in various different ways in different environments, and this is what helped them to increase their diversity. To capture energy, these guys relied on basically every method available. Now, you guys remember when we talked about autotrophs, we were talking about organisms that make their own food, and heterotrophs, organisms that get their food by eating other organisms. And we talked about within the autotrophs, even within the producers, we had different methods of making that energy. So we had photoautotrophs, they would use light in order to make their energy. We had chemoautotrophs, which would use chemicals to make their energy. And we would have within the heterotrophs, basically just consumers and decomposers and things like that, scavengers. So these guys were basically everything. They were heterotrophs. They would go along consuming other organisms. These are heterotrophic bacteria. They were photoheterotrophs, which are interesting. Photoheterotrophs were able to perform both photosynthesis and they could eat stuff, which is cool. They were like consumers and producers rolled into one. And there were photoautotrophs, which we know of. These guys perform photosynthesis. And then there were chemoautotrophs that used chemicals to make sugars, like those hydrothermal vent bacteria that we talked about at the beginning of the year. So they basically went out to the environment and they were like, look, I'll take basically any resource, anything I can use to turn it into energy to survive. Now, of course, that was how they collected energy, but how did they actually metabolize it? Because, this is a long time ago. the cellular respiration process that we know of today wasn't always shared by all organisms, and a lot of this stuff was pretty new. So there was like a large amount of diversity back then and today. But there was more diversity back then because at that point the atmosphere was still switching from methane and carbon dioxide to oxygen. And so a lot of organisms didn't just rely on oxygen like they do today. Actually, very few of them were what we call obligate aerobes, which means that they required oxygen. They did something very much like our modern cell respiration, where they took in oxygen and they, broke down sugars and produced carbon dioxide. But then there were also like a ton of obligate anaerobes, and those were organisms that they couldn't use oxygen. Oxygen was actually deadly poison to them; it would kill them. And so instead of being in oxygen, they would live deep within the mud or in the soil or whatever to get away from any oxygen that was present. And they would actually just rely on that fermentation process we talked about. So like lactic acid fermentation or ethanol fermentation, the same process that today we use to make beverages and all sorts of things. Back then, that was the only way that they were able to survive. And then there were also facultative aerobes, and facultative were able to switch between the two, though oftentimes they preferred being in an anaerobic situation. They could handle oxygen if necessary. Some people think that they represent sort of a transitional group between the obligate anaerobes and the obligate aerobes, but not really quite sure about that. So at that time, sex hadn't even evolved, which is a crazy thing to think about, but there was no such thing as sexual reproduction. So the bacteria reproduced using mitosis. Like we learned about, they would double their DNA, just like we do in the S phase of our cell cycle, and then they would just split in half, use that cytokinesis and all that stuff, and they were good to go. But as we know, mitosis creates two identical cells out of a single cell, and that's not great if you're looking for genetic diversity. So genetic diversity was pretty important in their populations because without it, if the environment started changing, they wouldn't be able to adapt without diversity. So to counteract this, they evolved something called conjugation, which meant that they would extend a little narrow tube between two bacteria, and that narrow tube would actually transfer some of the DNA from one bacteria to the other. And that DNA was very important DNA. It would be DNA that was like would give that bacteria the ability to survive in certain environmental conditions or give that bacteria sort of an edge in its fitness. And so what's really cool is that a bacteria would be like, hey, I'm going to make a bunch of copies of this very useful gene, and then they would just pass it out to their friends and be like, hey, everybody, who needs this like heat-resistant gene? And everyone would be like, I do, and they would pass it along using that tube and exchange genetic information that way. So that's how they got their genetic diversity. They also had a lot of mutations because mitosis can allow your population to grow very rapidly. It can happen a lot faster than meiosis. And because of that, a lot of mistakes can be made when you're copying your DNA over and over again. And that can allow for a couple of beneficial mutations to arise. So bacteria would have to basically survive in a changing environment every day. Earth was not stable, and it changed constantly. So in order to counteract that, some bacteria would find themselves, maybe they lived in the water. Well, suddenly that water was gone the next day. The bacteria evolved a way to be able to kind of survive in those really intense changes. And this survival basically meant that they would lock themselves into a shell. They would form a very thick wall around their DNA, and they would just stay in stasis for a really long time. This state is known as an endospore, and a lot of microorganisms living in extreme environments can do something like this. If you're interested and you have a moment, look up something called a tardigrade, Tardigrade, I believe. Tardigrades are also known as water bears, and they have this amazing process. It's kind of like the endospore, where they can change all of their, they can like alter a lot of their body functions so that they can survive for an incredible long period of time in extreme heat and extreme cold. I think they like replace most of the liquid in their body with sugar. I don't really know how it works, but it's amazing, and you should definitely check it out. So prokaryotes today are the most abundant form of life on the planet. A lot of people say because they evolved first, they're like primitive or they're not as evolved as we are and all that stuff. That is a view that assumes that there's progress in evolution, and there isn't progress in evolution. Evolution basically just goes with the forms that work, and prokaryotes worked really well. They still work really well. They outnumber us, millions and millions to one, and they're doing pretty well for themselves. They're really important. The Earth would not continue to function, or life would not continue to function, or at least not well, without these prokaryotes. They are consumers, they're producers, decomposers, they fix nitrogen in the atmosphere for us. They are absolutely essential for our survival. And they were the ones that originally created our oxygen-rich atmosphere. We were always like, oh, thank goodness for the plants for making oxygen. Well, yeah, but before the plants even evolved, bacteria were doing it. They help us to clean up oil spills by eating the oil, and they make yummy, amazing things like yogurt, which helps us in our health. So bacteria are really vital. It's also true that your body is host to thousands and thousands of species of bacteria, each of which does something really crucial and vital. Your intestines, for example, your gut flora, as we call them, you wouldn't be able to digest your food well or really survive without bacteria living inside of you. So pretty great that they're around. So, but in addition to being protectors, some bacteria can become kind of dangerous, and there are bacteria that are pathogens, which are disease-causing agents. So they can release damaging toxins or chemicals. They can eat through tissues, and they can cause a lot of distress and even death. So there are all sorts of diseases caused by bacteria, like Lyme's disease, tuberculosis, and strep throat. And we combat these by trying to reduce their numbers. So we try to use disinfectants. We try to slow down their ability to grow and to reproduce by using cold food storage. We process our food, so we cook it, and that helps to denature a lot of the bacteria and to kill them, or we'll sterilize them or use antibiotics. So there's lots of ways that we can combat these bacteria. Now, sadly, oftentimes when we combat the bad bacteria, we end up killing a lot of the good bacteria as well. And that's just a really difficult trade-off that is hard to find a way around. One of the most crucial problems that we're facing today is that bacteria are so good at evolving quickly because of that high mutation rate and because their generations are so short. They can go through like thousands of generations in a day, which means they can evolve at a rate that we can't even dream of. And since they can also pass on any kind of resistance that they evolve or that they adapt to other bacteria, without having to go through the process of sexual reproduction, they can develop resistance to our defense mechanisms very quickly. One of the big problems that we're facing right now is they are developing resistance to antibiotics, which is our number one way of treating really dangerous bacteria. Antibiotics are agents that mess chemically and directly with bacteria, so it can destroy their peptidoglycan wall, it can interrupt their metabolism, it can stop their reproduction. There's all sorts of things antibiotics can do. But bacteria can evolve through natural selection, and basically the individuals that are able to have mutations to get around the antibiotics survive and pass that on to the next generation. So they can evolve resistance and they can pass it on. The fear is that eventually some bacteria will evolve resistant to multiple or all antibiotics and become a superbug. And that isn't just science fiction. I mean, there's a distinct possibility of that happening. If that happened and people were infected with the superbug, it would be like a pandemic. It would travel around the world and many, many, many people would die, and it would be very difficult to treat. So that's what we're facing right now. It's a very difficult thing, and they're evolving so quickly that we might not be able to keep up with the antibiotics fast enough to keep them at bay. So that's a scary thought right now, but it's something that scientists are working on. We're actually, there's some really interesting research going on trying to use bacteriophages, which are viruses that infect only bacteria, to try to stop it. So it's kind of funny to think that we might try to use viruses to defeat another pathogen. So we've talked about bacteria, but what about archaeans? Because there's a second type of eukaryote, as you guys, or excuse me, prokaryote, as you guys remember. There are two prokaryotic kingdoms, eubacteria and archaic bacteria. So what are archaeans? Well, they evolved not too long, geologically speaking. I mean, it was several billion years, but not too long after eubacteria evolved. And archaeobacteria are very similar. They're single-celled. They have no nucleus or membrane-bound organelles, But archaeans were interesting because they adapted to environments that many bacteria couldn't adapt to. So they became what we call extremophiles. They loved the really extreme temperatures, like like thermophiles. They liked the super salty environments that would kill a lot of organisms. Those were called halophiles. And then they were extremely acidic environments, and those are acid phials. So archaeans would just go to places where no other organism would even try to think to survive, and they would do it. And before we really knew about archaeans, a lot of people, because they shared many similar traits to bacteria, or eubacteria, a lot of people thought that archaeobacteria were the same thing as eubacteria. But this scientist down here, whose name I'm blanking on right now. I'll put it in the description later. He was the one to discover that actually archaeobacteria were a different thing. And looking at the DNA markers within archaeobacteria, people actually realized that, hey, the DNA in archaeobacteria is way closer to eukaryotes, so eukaryotic cells, cells with a nucleus and membrane-bound organelles, our cells, than they were to prokaryotes. And so it turned out that, yeah, our ancestors were these extremophiles, these different bacteria that evolved into the third domain of life, and eventually led to us. So that's pretty incredible that out of all of these like chemical interactions and the crazy conditions of early Earth, we got this amazing evolution of these prokaryotes that diversified, and eventually the ones that you would least expect led to the creation of eukaryotic cells, and then to you and me, which is amazing. I mean, it's incredible to think that the environment that we know today was first populated and basically designed and created by the survival needs of bacteria. It's just amazing to think about. Anyway, I hope you've enjoyed this, and we're going to go ahead and, of course, after this, move into the eukaryotic kingdoms in more detail. But before leaving these behind, I just want you to think about what was the Earth like way back then, and what do you think about all of this coming from bacteria and the fact that they're still so crucial today? It's just amazing. All right, guys, have a wonderful day. Stay healthy, stay safe, and hope your bacteria stay in balance.